Hi all, here is a video I've prepared to help you with the Unit 3 homework. I hope with this video I can provide you and your group with the tools you need to tackle the nuclease digest problem. First, we're going to talk about chromatin structure. I'll go briefly into histone modifications, and then I will walk you through the in-lecture example of a nuclease digest. I'm very excited to be sharing this with you. Chromatin biology is what I study here at OSU. So if you ever find yourself wanting to learn more about this branch of biology, please reach out to me or to my advisor, Dr. Michael Freitag. I want to give a brief overview of chromatin structure. As you've already learned in the lectures and the course notes, chromatin is a combination of DNA and proteins. In fact, before we even knew that nucleic acids were the genetic material of a cell, scientists isolated and deconstructed chromosomes, identifying the building blocks to be comprised of about 40% nucleic acids and 60% protein by weight. I think of this as the branching point between the disciplines of biochemistry and genetics. Biochemists saw this number and thought, chromosomes are mostly protein. Protein is comprised of 20 or 21 amino acids. This macromolecule really has the possibility to carry a great deal of genetic information. And of course, geneticists thought that the nucleic acid was the genetic material, and now after a few really foundational experiments, we know that the geneticists were correct. So how is DNA packed into chromatin? Well, the individual building block of chromatin is the nucleosome. The nucleosome is an octamer of 4 histone proteins: histone H2A, histone H2B, histone H3, which is my personal favorite, and histone H4, all wrapped twice around the double helix of DNA. When this octamer of histone proteins are incubated with DNA, they form the beads on a string structure, which is also called the 11 nanometer fiber, and it's named 11 nanometers for the width of the fiber of the DNA in complex with the core histones. With the addition of another histone protein, histone H1, which is not part of the core nucleosome, the 11 nanometer fiber condenses into what's known as the 30 nanometer chromatin fiber. As we get to these higher order structures up here, less and less is known about the state of the compaction of the chromatin, and it's an ongoing area of research today. I do want to point out one really important distinction to add to the topic of chromatin structure. So we've covered how chromatin is made up of DNA and protein, but chromatin is also comprised of RNA, and the degree to which RNA plays a role in chromatin structure is going to vary greatly among organisms. For example, let's re-examine the phenomena of chromosome silencing in females who possess 2 copies of the X chromosomes in each of their cells. I hope you have an appreciation for this at this point, but you may not get there until the end of this course. Gene regulation is a very tightly monitored process. So to prevent double dosage of the genes on the X chromosome, one of these chromosomes is converted in its entirety to heterochromatin, meaning that the genes on this chromosome are no longer expressed. The chromosome that gets silenced is chosen at random, meaning the result of the X chromosome inactivation, pardon me. Sorry about that interruption. My son is watching Encanto on the tablet next to me. I hope there'll be no more interruptions.
I love you, Mama.
Oh, thank you, sweetie.
I love you.
Thank you, love. Okay, so I'm gonna restart this slide over. I apologize for any confusion that may cause. All right, so let's re-examine the phenomena of X chromosome silencing in females who possess 2 copies of the X chromosomes in each of their cells. To prevent the double dosage of genes on the X chromosome, one of these is converted to heterochromatin. The chromosome that gets silenced is chosen at random, meaning all the result of X chromosome inactivation or bar body formation as some people call it, is a mosaic of genes expressed from the X chromosome. So let's talk about what that looks like. A classic example of X inactivation is seen in cats. If a female cat is heterozygous for black and tan alleles of a coat color gene that's found on the X chromosome, she will inactivate her two X chromosomes at random in different cells during development. And so the result of this is a tortoiseshell cat pattern made up of alternating patches of black and tan fur. The black patches come from groups of cells in which the X with the black allele is active, while the tan patches come from cells in which the X with the tan allele is active. This mosaic is easy to see for a tortoiseshell cat, but guess what? This is also true for human females. There are mosaics for any genes that are present in different alleles on their two X chromosomes. Okay, that's it for genetics and this little lecture, I promise. So back to cell biology. I want to share with you an example of X inactivation. First, I'd like you to take a few seconds to identify what type of microscopy was used to capture this image. Technically, this was captured on a confocal microscope, but that is a subset of emission light microscopy. So I hope you guessed ELM. This image was captured using a specific technique commonly used in cell biology. It's abbreviated as FISH for fluorescent in situ hybridization. FISH is a technique relying on specific base pairing of a fluorescent probe with a particular DNA or RNA sequence. In this case, the researchers labeled EXIST, a gene carrying the instructions of a non-coding RNA found on the X chromosome. What is the purpose of the EXIST non-coding RNA, you may ask? Well, it's important for chromosome inactivation. In the image here, we see the nucleus labeled in blue, the EXIST gene on DNA labeled in yellow. In this image, we can see two distinct EXIST DNA loci because this is a female mouse cell, and each female has two X chromosomes and therefore two EXIST genes. But if you'll notice, the EXIST RNA transcript shown in red only localizes to one of these chromosomes. The chromosome labeled as XI here, the one with the EXIST RNA cluster, is the inactivated X chromosome. EXIST RNA recruits many other chromatin modifying enzymes, and together these two processes result in the formation of heterochromatin and X inactivation. I'm sharing this with you as a reminder that depending on the organism, RNA can actually play a really important role in the chromatin structure. And in this case, RNA is playing a role in heterochromatin formation in female mice. In addition to RNA, the state of the chemical modifications of histones can affect chromatin structure. For example, in euchromatin, shown here in green, we commonly see histone H3 lysine 4, or H3K4, methylated. Regions of the genome that are covered with this histone modification are generally considered to be less compact, and that's been validated in some organisms, and therefore we say they're more open for RNA polymerase to initiate gene expression. In contrast, in heterochromatin, we often see histone H3 modified with methyls either on lysine 9 or lysine 27, and this chromatin is typically portrayed as more compact. Regardless of the compaction of the chromatin, the blueprint of DNA bound to the nucleosome is still about the same. So I think that leads us nicely into our discussion of our micrococcal nuclease experiment. Micrococcal nuclease is an enzyme that cuts exposed DNA And so therefore it can be used to determine what DNA is tightly bound to proteins and thus protected from cutting by the nuclease. An important concept with this technique is that with a long enough exposure to the micrococcal nuclease, all exposed DNA will be destroyed. I want to include this as an important distinction. Last week's homework relied on a polyacrylamide gel to separate membrane-bound proteins based on size. And for Unit 3, this time we're going to want to separate DNA molecules based on size, and we will do this with an agarose gel. Agarose is a compound purified from seaweed. I'm not sure how many of you have taken a microbiology course, but you may or may not have poured media into Petri plates for some sort of microbial growth. You likely used a substance called agar, and this gives the media its characteristic jello-like consistency. Agarose is a more purified and way more expensive derivative of seaweed than just agar. And agarose is also an appropriate media to separate DNA molecules by their size. Just as was true last week, when we talk about a polyacrylamide gel, in an agarose gel, DNA molecules will migrate. Smaller DNA molecules will migrate faster, shown here, and larger DNA molecules will migrate slower. We always run a molecular weight standard, which is likely lane 1 in this image here. And this gives us a standard to compare our fragments to. So, to approach this problem, I want you to break it up into a couple different steps. First, think about what proteins are associated with the DNA at each step. Second, ask yourself, what effect will the nuclease digestion have on the overall structure? And will the length of the digestion have an effect on what you draw? And finally, you want to think about what you end up with after step 3 in each treatment. This is going to be the naked DNA, and this will be what you load into your gel. Alright, so here's the problem we're going to work on together. When researchers were first trying to understand the structure of chromatin, they tested how much of the DNA was protected from nuclease, which degrades DNA not attached to histones. To produce the figure, a 240 base pair fragment of chromatin was subject to digestion by micrococcal nuclease for the indicated time. The DNA was then separated from the histones, run on a gel, and viewed. Based on this data, what is the length of DNA represented on the nucleosome core? So we're going to approach this problem just as I'd like you to do for the homework. I've created this table, which is similar to what you have in your homework and not exactly the same, and I added an extra lane for undigested chromatin, and this is just to give you a starting point for your homework. So our starting chromatin in all of the treatments is going to look the same, and that's also true with your homework. For this particular example, you're told that you only have 240 base pairs of chromatin. This is a really small fragment. In your homework, expect to draw more than 2 nucleosomes. Maybe draw 4 or 5. It'll probably be easier for you if you do it that way anyway. So our starting point is the same. We have DNA in complex with our nucleosome particles. If we don't incubate this chromatin with nuclease, guess what? We expect it to look exactly the same. This is not shown on the gel to the left, but if it was, we would expect to see it somewhere above the 200 base pair molecular weight standard. Upon treatment with nuclease, we start to see different populations of chromatin fragments arise. After 15 seconds, some are very large, but we start to see populations as low as 160 base pairs. The way that I would indicate that is there's some that's still completely undigested, while some is starting to be broken down. By 30 seconds, we don't really see any more of the super large fragments above 200 base pairs. and we start to shift towards a smaller size. I've indicated this by showing slightly more digested fragments. I'm going to rush through the next 3 time points, 1 minute, 2 minutes, and 5 minutes, because I think my comments can be made about all of them. We're shifting in that time period from populations of chromatin comprised of larger fragments to chromatin comprised of smaller fragments. But in each of these 3 time points, we have populations indicating we have multiple sized fragments. And I've indicated that here with these nucleosome particles. Finally, after a complete digest, we see only 1 population of chromatin left, with the exposed DNA completely digested. Now, to extrapolate what the naked DNA would look like after the dissociation of histoproteins with a salt solution, this is what we expect to see. Generally speaking, with the addition of additional time with the nucleus digest, we start with larger fragments at 240 base pairs, and as time goes on, we start to see less and less of the larger fragments and more presence of the smaller fragments, which, based on the gel over here, reside at about 140 to 150 base pairs. The completely digested chromatin at 10 minutes, therefore the answer is going to be D, 140, but at least based on this gel here, is between 130 and 150 base pairs. Depending on the organism, the blueprint of DNA around a nucleosome varies slightly, but typically it's between 140 and 150 base pairs. Okay, so hopefully that example made sense to you. Now let's add a level of complexity. So this is the picture that Maka, Dr. Franco, drew last term, and I shared it with you yesterday in the announcements. I'm going to help you walk through this. First, I'm going to talk to you about chromatin assembly, and then I'll talk a little bit about the experimental protocol, and finally leading to what the chromatin actually looks like at the end of this. Okay.
Mama, look what I'm doing.
All right. So by now, I hope you know that naked DNA is complexed with the core histones, histone H2A, H2B, H3, and H4. And together this forms the nucleosome. Each nucleosome is comprised of roughly 140 to 150 base pairs of DNA. Multiple nucleosomes form the 11 nanometer fiber, also called the beads on a string model. When we add histone H1, it binds to the linker region between 2 nucleosomes, right here, and has a blueprint of about 60 base pairs, meaning that a nucleosome complexed with H1 takes up about 200 base pairs of DNA. Once H1 is added, we know that chromatin starts to organize into the 30 nanometer fiber, and Maka did a great job of indicating here that this organization and the assembly of higher order... One second. Sorry about that. As I was saying, Maka does a great job of indicating here that the assembly of higher order structures is still an active area of research today. All right, so let's go to the left, talk about the experiment. In this experiment, we're actually starting at the bottom, right? We are starting with this 30 nanometer fiber, as indicated by your homework. The undigested chromatin all had the structure of the 30 nanometer fiber. We first treat this with a weak salt solution, and that's going to dissociate histone H1. So in this case, we're taking a step backwards. We're moving from the 30 nanometer fiber, dissociating H1, and the resulting chromatin structure is going to look like this 11 nanometer fiber. If we treat an 11 nanometer fiber with nuclease for a short time, 30 seconds, we end up with a population of fragments of chromatin. And once we remove the core histones and run the naked DNA on a gel, we would expect to see a variety of fragments of DNA. With a long nuclease incubation, we expect all exposed DNA to be digested, and therefore uniform fragments of DNA about 140 base pairs in length after we use salt to remove the histones. I hope that the in-class example and this image helped clear up some things. As always, please don't hesitate to reach out if you have more questions. And thank you for your patience as my son and I are in COVID quarantine in the same space. So I appreciate your patience. Please reach out if you have more questions. Thanks.